Шановні присутні, розпочинаємо роботу нашого наукового семінару. Як ви знаєте, участь в цьому семінарі приймаємо ми тут наживо і наші колеги в різних містах України. Сьогодні, я знаю, до нас приєдналися Львів, Харків, Івано-Франківськ, моя рідна Полтава і Миколаїв. Ну, розумієте, фізика не має кордонів, як будь-яка наука. Але все ж таки кордони сьогодні існують. Це кордон з агресором, який вже майже зробив рабів в своїй країні. Хотів би зробити рабів в нашій країні. Ну, наприклад, що вони роблять? Забороняють користуватися інтернетом. От як ви думаєте, якби тут хтось нам сказав, що інтернет – це не для нас, що б ми йому відповіли? Ну, те, що українці відповідають росіянам вже 4 роки. Нас народили вільними людьми, і Україна є і буде вільною. А вільна Україна для нас з вами що це? Ну, для нас, викладачів, вільна Україна – це вільно розповідати нашим студентам про те, яку спеціальність вони обрали, розповідати про новітні досягнення в фізиці, розповідати про контакти, які існують у нас з нашими колегами за кордоном. І кожен з наших, а що для студента таке вільна Україна? А вільна Україна – це отримати прекрасні знання, працювати на нашу рідну Україну. А якщо людина захоче працювати за кордоном, будь ласка. Їх знання дозволяють це робити на рівні з випускниками університетів інших європейських країн. Я знаю наших професорів, учні яких працюють в багатьох країнах. Для прикладу я скажу про своїх докторів та кандидатів наук. От, наприклад, сьогодні працюють мої учні, доктори та кандидати працюють в Трієсті, в Генуї, в Відні, в Іспанії, в країні Басків, в університеті. Працюють в Парижі, працюють в Будапешті, в Познані, от в Оксфорді працюють, в Окриджі. І це мій приклад. Я не збирав такі приклади по всьому факультету, але повірте, ми готуємо фахівців таких, які достойні працювати в Європі. І один із таких моментів, які нам дозволяють це робити, це наші контакти з нашими колегами за кордоном. Нам особливо приємно мати такі контакти, коли наші колеги представляють ще країни, які підтримують нас в нашій боротьбі. Безумовно, Велика Британія – це одна із тих найкращих наших помічників в нашій боротьбі за незалежність. І я дуже радий сьогодні представити вам професора Ірландського університету Чарльза Кокелла. Тематика його доповіді близька нам. Це тематика, яка стосується і астрономії, і медичної фізики. Принаймні з 3 кафедр, які розвиваються у нас на факультеті, з тих кафедр, 10 кафедр, 3 кафедри причетні до вашої доповіді, шановний Чарльз, це кафедра астрономії, кафедра молекулярної фізики, кафедра фізики функціональних матеріалів, на яких розглядається питання медичної фізики. Я дуже радий надати вам слово, шановний Чарльз, і будемо раді вас бачити в наступні роки. Дякую. Вам час 40-50 хвилин.
Дякую. Дякую, друзі. Друзі, я дуже радий бути тут в Києві, в університеті знову. Моя українська мова не дуже чудова. Я вивчаю українську мову в Единбурзі. По-перше, я хочу подякувати фонду президента України. Для мене велика честь отримати підтримку за фонду. Сьогодні я буду говорити англійською. Okay. Thank you very much, and it's great to be here in the physics faculty. Today I want to talk a little bit about some research that I do in my laboratory. I am a biologist. I am in a Department of Physics and Astronomy in Edinburgh because I call myself an astrobiologist. I'm interested in connecting astronomy with biology. So the university, with their wonderful bureaucratic systems, decided that because I'm an astrobiologist, I should be in a physics and astronomy department. But I'm actually trained as a biologist. What I'd like to do today is to talk about some of my research that I think connects with physics and chemistry, in particular understanding the limits of life. And I'll talk a little bit at the end of this lecture about applications to space exploration and space missions that have a sort of physics content. I should say I'm here for a month. As I said, very kindly supported by the fund of the President of Ukraine. I'm here till mid-June. I've been here for a couple of weeks. And if anyone is interested in talking about astrobiology, life in extreme space missions, I'm always interested in talking about these subjects. So please get in touch. Ludmila can make contact and can have some discussion. I should say I also already gave a short introduction lecture to the students here, and it was a really excellent and wonderful discussion that we had. So thank you to them. Today is more of a scientific seminar than a student lecture. So I'm going to talk about the shape and limits of the biosphere. I'm then going to explain some work on how chemical ions can establish limits to life. Then I'm going to talk about some work we did studying life after asteroid impacts. You can consider this to be a sort of physically extreme condition throughout the history of Earth. And then I'm going to talk about microbes in space biotechnology, which may seem to be a completely disconnected topic, but is quite similar to what I'm going to talk about before. Now, this is something that may be obvious to you as physicists. In fact, it may be obvious to any scientist, but I still personally find this remarkable, that biological sciences is currently confined to a single planetary object in the entire observable universe. So physicists, for example, they can measure the cosmic background, microwave background radiation. They can tell you something about the very beginnings of the universe, and they can map the structure of the universe. They can tell you about how the universe has changed over 13.5 billion years. And chemists too, they can take a telescope, they can measure the spectra of distant galaxies that were formed hundreds of millions of years, so really quite soon after the Big Bang. They can tell you something about the chemistry of the universe across space and time. If you talk to a biologist like me, everything I say about biology applies to one object in the entire observable universe. We only know of one biosphere. And of course, this is not a photograph in the bottom left, but that's sort of the Milky Way, roughly where you are. And on the right-hand side, this is a photograph taken by Voyager 1, 6 billion kilometers from Earth. And that tiny blue dot where that arrow is pointing is planet Earth, taken by Voyager 1. So on that dot is biology, physiology, immunology, neurobiology, biochemistry, every biological science. And I think that's an amazing thing. It's an amazing state, you could say, of primitive knowledge. And a fundamental question for biologists, which requires collaboration with physicists, is are there other instances of life, of this replicating, evolving matter that we call life? Are there other instances of this matter elsewhere in the universe? Or is, in fact, is it empirically the case that this is the only planet, at least in our near galactic neighborhood, that has biology? That's a fundamentally interesting question. And it's often put to the public as, are we alone in the universe? There are sort of more popular ways of asking this question, but the fundamental science question is biology, is replicating evolving matter isolated to a single planet, or can we find examples of this type of material elsewhere in the universe? This, to me, is the central challenge of biology. Now, the other interesting thing about biology is even on the one planet where we know it exists, it's extremely limited. So here is a very basic cross-section of Earth, the geological structure of the Earth, the core and the mantle and the crust. And you could ask yourself, where is biology on this planet? So if I take away all the geology and I leave just the biology, that's the biosphere. And I've increased the thickness of that line by about 5 times, because otherwise you can't even see it on the slide. The biosphere is about 5 to 10 kilometers thick, and it constitutes about 0.3% of the volume of the Earth. Even on the one object in the observable universe that we know can support life, the biosphere is very thin. And in my laboratory, I'm interested in understanding what these limits to life are. This is what the biosphere looks like. It exists, that intersection between the blue and the brown is the surface of Earth. Biosphere is beneath the surface and up into the atmosphere. And when I talk about the biosphere, I mean actively replicating life. You can find microorganisms 70 kilometers altitude in the stratosphere drifting around. They're not actively replicating. They're desiccated or they are frozen. So the actively replicating biosphere is this thin layer of life. Why is this the case? Well, the reasons are physical, that they're rooted in physics. If you drill into the subsurface of the Earth, you will find there's an increase in temperature, the geothermal gradient. Temperature of the Earth increases as you go down into the ground. This is the surface of the Earth. You can see in the middle there, and that red line on the right-hand side at 122, this is the currently known upper temperature limit for life. This may go higher in a few years, but at the moment it's 122. And this sets the limit for life. And the depth of that limit is about 5 kilometers, or roughly thereabouts. The geothermal gradient varies in different parts of the Earth, but it's roughly about 5 kilometers. That's the depth of the biosphere. If you go up into the atmosphere, the temperature at which, like the minimum current temperature in the literature for the replication of microbes is minus 25 degrees Celsius. That may go lower, but it's roughly about this. And the altitude at which you find that temperature is about 5 kilometers high. But in reality, the height of replicating life is a lot lower because of desiccation, ultraviolet radiation, lack of nutrients. So we think that actively reproducing life is probably something like 2 kilometers altitude in the atmosphere. So the thickness of the biosphere is the biosphere is very thin. And Volodymyr Vernadsky understood this. And I should say, perhaps you're aware that this year is a very special year because it's the 100th anniversary of Vernadsky's book, The Biosphere, which he published in 1926. Vernadsky, of course, being the first president of the National Academy of Sciences of Ukraine. And in 1926, in his book, he described the biosphere as being an envelope of life. So he understood this 100 years ago, but he did not understand the physical reasons for why the biosphere was so thin. We now have a better understanding of the limits of life, and we can draw diagrams like this, and we understand why life is so thin. So anyway, what I wanted just to explain to you is that there is one planet so far in the observable universe that has life, and even on this planet, biology is a thin layer of replicating, evolving matter just beneath the surface and in the atmosphere. And the question is, why is this the case and what sort of limits might impose themselves on life. So the first thing I want to talk about, just transitioning now to some research and some science, is what other things might limit life in the biosphere. And a few years ago, we went to a mine, an active mine in the UK called the Bulby Mine, which you can see in the top left-hand side. It's on the eastern coast of the UK. It's an active mine, it's 1 kilometer deep. And in this mine, there is a laboratory on the right-hand side where scientists are looking for dark matter. So maybe you as physicists and astronomers will be more familiar with this research than I am, but it's also the perfect place to look for microorganisms in extreme conditions. And this mine is located in what was once an ancient sea, the Zechstein Sea in the bottom left, which was formed 250 million years ago, and it contains layers of salts, layers of different ions that were laid down 250 million years ago. And in the right-hand side, in the bottom right-hand side, you can see that we can go into this mine and we can collect different types of fluids that have worked their way through these salts and produced these concentrated liquids that contain different concentrations of ions. And we can have a look to see which microorganisms have grown in these fluids and which fluids can support biology. So what we're looking at here is the depth of the biosphere. So you remember that 5 kilometer deep temperature gradient. Question is, can ions limit the depth of life in the biosphere independent of temperature? So we're looking at the microbes growing in these different conditions. Now, just a little bit of an introduction to the effects of ions on life. Maybe some of you are familiar with water activity. You can measure the availability of liquid water to life on a scale called water activity. And in formal terms, the water activity is the vapor pressure above a solution, a liquid solution, as a ratio of the vapor pressure above pure water. And this can also be described as the relative humidity, if you express this as a percentage. 0 is completely dry, 1 is distilled water. In between 0 and 1, we have different water activities depending on the amount, the concentration of ions, and those water activities set the limits for life. So you see this red line near 0.6. This is the currently defined limit to life, currently defined water activity limit to life. Lower than that, it's not possible for life to replicate. Above that limit, life can replicate. So magnesium chloride is about 0.35. In a concentrated 3 molar magnesium chloride concentration, life cannot replicate. And on the right-hand side, you can see sodium chloride, magnesium sulfate, and at the bottom of that scale, various organisms that can grow in these salty conditions. But in general, anything below about 0.6 water activity, there isn't sufficient water for life to be able to replicate. At least that's the currently defined limit to life. So if you go down into the mine and you collect different fluids, what you can see is in this diagram that is not very useful, but I've highlighted in there the water activity of 4 different brines formed from the fluids that are passing through these ancient salts. And if you can see, the first 3 of those have a water activity of about 0.7, which is above the limit for life. And in those brines, we find that life can replicate and grow, which is what we would expect. In that 4th one, you can see the water activity is below 0.6. And we would predict that in this fluid, the water activity would be too low for life to be able to exist in these brines. There's another interesting phenomenon, is that certain salts, certain ions, cause disorder in molecules. Magnesium chloride, for instance, causes molecules to unfold. And this is called chaotropicity. So it's a measure of disorder. And all I want to show you in this slide is that in that small red box, this is a laboratory method for measuring the disordering tendency of ions. That this magnesium chloride brine here, which contains high concentrations of magnesium and chloride ions, is extremely disordered. So as well as a low water activity, we also have a disordering effect on molecules. Okay, so this is a brine that is disordering to life. So what does this mean? It means that in this particular brine with high concentrations of magnesium and chlorine, microorganisms are not able to replicate. And in this diagram, what you're looking at is two salts on the right-hand side, one of which is capable of supporting life. And the red boxes on the very right-hand side of this diagram are samples of this brine we've introduced different types of microbes into to see if they will grow. And put simply, all of these red boxes show you that this brine will not support life. So what we have here is a fluid that cannot support the replication of life. Why is this interesting? Well, because most of us tend to think that where there is water, there tends to be life. What we have here is an example of liquid water, but it cannot support life. And this is at 1 kilometer depth. So it shows that our biosphere, when it is normally restricted by temperature, the physical constraint of temperature is about 5 kilometers depth. But with certain ions in a planetary environment, there can be a chemical limit to life at much shallower depths. Even at 1 kilometer depth, if we have magnesium and chloride ions, which are quite common on Earth, we can find a depth at which life is restricted much shallower depths. So this is an example of ionic limits to the biosphere. I don't know if I can get this to close down. Maybe not. I can move it. No. Okay, I won't try and do that because I'll mess it up. Anyway, it says an ionic limit to the biosphere. And this is an example of ions limiting biosphere. Ultimately, this is a physical effect. It's ions interacting with molecules in a cell, denying them the use of water and unfolding proteins and preventing active operation of these enzymes. Now, I have an interest in space exploration, so I'm interested in thinking about what conditions could limit life if it existed on other planetary bodies, casting our mind back to this question of, are there instances of life elsewhere in the universe? One question we could ask ourselves is, how would chemistry and physics on other planets limit life, limit the possibilities of life? One interesting planet is Mars. We know that in its early history, Mars had abundant liquid water. So on the left-hand side is an image of Mars in the present day and an artist's impression of what it might look like about 4 billion years ago. We know this is empirically the case because we have evidence for ancient liquid water on Mars. In the top right-hand side is an ancient river flowing into a crater where there is a delta that formed in an ancient lake. And right now, NASA's Perseverance rover is inside this crater studying these ancient lake deposits. So this is not speculation. We know that there was water on early Mars. And in the bottom right-hand side is calcium sulfate veins, which formed from water evaporating and depositing ancient salts on the surface of Mars. So the question you could ask yourself is the situation on Mars the same as on Earth? Would life be limited by ionic strength and the availability of water, or is the chemistry of Mars different? Would this lead to different conclusions? So what you can do is you can make artificial Martian brines or salts in the laboratory based on data that's been sent back by rovers on the surface of Mars. We can make Martian fluids. We have enough data to do this. And in this experiment, what we've done is we've made a variety of dilute and concentrated Martian brines, and we've introduced microorganisms into these brines to see whether they will support life. And on the left-hand side are just 5 of these brines and information on the different types of bacteria growing in these brines. But I'm not going to talk any more about this. This is sort of biological detail you don't need to know. Just want to show you on the right-hand side is a sodium chloride brine that has this bright orange color. This is a typical brine on Earth. And on the left-hand side is a Martian brine that is made up of these sulfate salts that I'll talk about in a moment. And they both support life, but they both have very different types of microbial communities. Now, what's interesting about Mars is that the solutions of the salts on that planet are dominated by multivalent ions like magnesium and iron and sulfate, rather than sodium chloride, which is made up of monovalent ions, Na plus, Cl minus. So monovalent ions on Earth, like sodium chloride, dominate brines. Sulfates dominate brines on Mars. And this is a consequence of a different geological history. On Mars, the water disappeared as it became a desert planet. This water reacted with volcanic rocks, and it formed these sulfate-type brines on the surface of the planet. So what we could do is we could say, given data on the growth of microorganisms in these brines, does water activity predict the growth of microbes in these brines? So this rather complicated diagram, but actually it's quite simple. The data on it, the squares on this diagram, the green squares are places on this planet that support life, and the red squares at the bottom of the diagram are places on Earth that do not support life, including this example I gave in Bulby. And the red line here is the water activity limit for life. It's actually now slightly below 0.6, but when we published this data, it's just above 0.6. And what you'll see is on Earth, the green squares that can support life are above that limit of water activity. The red squares are below them, which is what we would expect. But interestingly, for the Mars brines, you will see on the right-hand side, there are some red circles. These are the Martian brines that are above the predicted lower water activity limit for life. In other words, water activity does not predict Martian brine habitability. And I should say that those lines with the black arrows represent different evaporative states of brine. So a dilute brine evaporating to a more concentrated brine, and the arrow is the movement between those two brines. So what's going on here? Why is it that water activity does not predict Martian habitability? Well, there's another interesting physical effect going on here, and that is that if you have a lot of ions that are multivalent, like magnesium 2+, iron 2+, and sulfate 2+, you end up with a high charge density in the fluid, a high ionic strength, as it's called. So this is not just about water availability. What we've got now is a problem with ions with high charges, which at high concentrations generate high charge density. In other words, high ionic strength. And ionic strength has the effect of disrupting biological molecules. If you surround DNA and other proteins with magnesium 2+ and sulfate 2+ and have lots of charges, multivalent charges, you will find that molecules are disrupted. And if we plot ionic strength on the x-axis against water activity, now you can see that all the Martian brines sit on the right-hand side of this high water activity of about 10 molar. In other words, ionic strength on Mars predicts the habitability of brines and not just water activity. Why is this interesting? Because it shows that different physics and chemistry of ions in different planetary environments can limit life. So on Earth, we as biologists routinely measure the water activity in fluids, and we define the limits to life based on water activity. And that is because we live on a planet where most of the brines, not all of them, because as I showed you before, there are some that have different ions, but most of the water bodies, including our oceans, are dominated by these monovalent ions. So water activity is the key physical limit. But on planets where you've had a different geological history and different ions have been leached out of rocks, water activity and ionic strength may define limits to life. And so we can see that the different histories, that the divergence of the history of these two planets leads to very different conclusions on the limits to life. This is interesting to a biologist because ionic strength is rarely discussed amongst biologists as a limit to life. And when we found this, it was interesting because it was a rare example of how a completely different physical parameter, charge density, can limit life in addition to temperature and pressure. And since then, and I won't talk about it, but if you're interested, my laboratory has also studied effects of salts on pressure. We've also looked at life under ammonia as ions and interactions of these different ions. And just to mention, one of the interesting things we found is that you can use ions like perchlorates that disorder molecules to reverse the effects of high pressure. So under high pressure, molecules will be squashed together. They reduce their volume. But if you add an ion like perchlorate that causes unfolding, it can reverse the effects of high pressure and improve the activity of a molecule. So sometimes these salts that cause disordering can have beneficial effects to life by reversing the effects of in some sense, too much ordering. Now, another thing that I'm interested in as a biologist is how has this biosphere been changed over time by radical physical events? And one of the most radical events that you can have on the planet is an asteroid or comet impact. This is the asteroid impact that we believe, and there's good evidence for this, was responsible for the end Cretaceous extinction that killed all non-avian dinosaurs and 75% of life about 66 million years ago. And the crater associated with this event, called the Chicxulub crater, is buried under the sea floor in Mexico on the right-hand side. And so I led a microbiology effort, which was part of a geology study of this crater. The geologists wanted to know, how did this event occur? What did it do to the atmosphere that caused such calamitous changes to the biosphere? I was interested in how has a catastrophic event like this affected that thin layer of the biosphere, the microbes underneath. So we went off and spent 3 months on this drilling rig in the Gulf of Mexico in the bottom right-hand side. This was an interesting experience. We ate fried chicken for about 3 months, which got very boring, but we got good material from this core. And first to say that these events are quite rare. We now have a knowledge from the cratering record in our solar system, also from the observations of asteroids, about how frequent these events are. This is just a graph showing you the energy on the x-axis released in megatons of TNT, the explosive TNT. This is a rather odd physical unit that is routinely used by impact geologists. It's just a historical thing. And then on the y-axis is the frequency of the impact. So this type of impact occurs about once every 100 million years. And then there are events that are less catastrophic but occur more regularly. So the Tunguska impact was not actually an impact. It was an object that came in, exploded above the ground, and it flattened about 2,000 square kilometers of forest in Siberia in 1908. And these sorts of events happen about, well, we predict about once every 500 to 1,000 years. So they're not so rare. So there's this estimation. That gray bar there is the transition from a local effect to a mass extinction on a global scale. And I'm interested in how do these events affect microorganisms. So what we can do is we can go and drill into this crater, which is what we did. And on the right-hand side in the top, you can see the drill core. And just to take you through this quickly, the pink part in the top right, that's the granite that comes up from the deep subsurface, from the crust of the earth, right? Granite. Yeah, I'm learning strange Ukrainian words in science over the last couple of weeks, exoplaneti and other things. So I'm beginning to understand how to say these things. But anyway, that's the granite that comes up from underground. The blue material is the material in the middle, and this is called swavite. And this is the material that was fractured by the asteroid impact. So you have to imagine, if you were standing there 66 million years ago, which would not have been a good idea, but if you'd been standing there, you would have seen a 10-kilometer diameter rock coming from space. It would have carved a hole in the ground, and the models suggest that it generated tsunami waves about 1 kilometer to 3 kilometers high that swept across the US. And those waves washed back fractured rock with pore spaces. And this swavite in the middle there This is this broken up rock, was washed back into the crater and filled the crater. And this catastrophe happened in about 40 minutes, so less than the length of this lecture. So it's a very rapid, extraordinary event. Now, what's interesting is that in this swavite, you can see at the top there, that first set of data is the number of bacteria in the core. And you can see this spike of bacteria. The next column is DNA, so the genetic material. And you can see these spikes of where there is an increase in microbes. And then on the far side, next to the core, you see this spike in porosity. Porosity being the measure of the pore spaces and the fractures in the rock. So what's going on here is that the asteroid impact has fractured the rock. It's created pore spaces, and through those pore spaces, fluid can move and feed the microorganisms and provide them with energy. So in other words, asteroid impacts are bad for dinosaurs, but they're good for microbes because they provide fractures through which fluid can flow and provide food and energy. And so this suggests that although we view these things as catastrophic, when you think of that biosphere that I mentioned earlier, let's just go back to here. For microorganisms, asteroid impacts will increase the surface area and the food available through the crust and may actually improve the conditions for life. So these may be beneficial for life rather than purely destructive. I'm an astrobiologist, so rarely do I do any science without thinking about space. It's just the way I think. So here's an application to space. If you go to Mars, you will find a very large number of impact craters. This is a map of Mars, and every single one of these dots, these yellow and red dots, is an asteroid or comet impact crater. And about 350,000 craters have now been mapped on the surface and subsurface of Mars. You might say, why are there so many? Because the impact flux on Earth and Mars have been the same over solar system history. Well, on Mars, there's no ocean and there's no plate tectonics. So most of these craters are preserved over a 4.5 billion year history. On Earth, there are about 300 well-characterized, well-understood craters, but most of the craters that have ever occurred on Earth have now long since disappeared. So what's this got to do with what I said before? Well, if you were looking for life on Mars, my suggestion would be that you should go and look in the fractures of craters. And on the right-hand side is a picture of Victoria crater taken from orbit, and just above it, an image taken by NASA's Curiosity rover, which drove up to the edge of that crater and looked over the edge. And there you can see this crater with lots of fractures and cracks in it. And when I look at photographs like that, I get really excited because I think if I was an astronaut and I had a rock hammer, I would climb down the side of that cliff and I would go and collect rocks in that crater and I would look in those fractures and see if there's life. And if I found no life, well, that wouldn't prove there's no life on Mars, but it would suggest to me very strongly that some of the most promising environments on Mars did not support life. So this is the way in which we can have, I guess, a more directed series of hypotheses about how to search for life on Mars, rather than just going and looking around and trying to find life everywhere. We use life on Earth to create good scientific hypotheses to look for life on Mars. And this is just one example of how impact craters can help the search for life on Mars. But here's just one final thought about this. I just showed you how deep in the Bulby mine we can have high concentrations of salts, and I showed you how Martian brines can limit life with ionic strength. So one of the arguments you could think of is if you increase the fractures in the rocks through Mars, this will improve the movement of these concentrated brines through the subsurface and could prevent life from growing there because it's too extreme, right? So the Martian brines find it easier to get into the subsurface and they can stop life. So did impact craters improve conditions for life on Mars, or did they make it worse? Well, we don't know. And the best way to test this hypothesis would be to go to Mars and drill into the subsurface and collect samples and see what the effects of impact fracturing were on the subsurface environment. Of course, it is very difficult to drill on Mars. It's very technically complicated, but there are some are engineers who have some ideas about how we might go and drill on Mars. So I would suggest picking some of these craters and drilling into them and looking for evidence of life. So this again is a connection between different physical principles. I think it's appropriate here in this faculty just to emphasize that everything I'm talking about are consequences on biology, but it's all rooted in physics, right? Ionic concentrations are to do with ions and the physical behavior of ions. Asteroid impacts are destructive simply because of kinetic energy. It's purely a physical process affecting biology. So underlying biology is always physics. Don't tell the biologists that. It's true. So the next thing I want to talk about, the last thing in my talk I want to draw attention to is how can we use a knowledge of biology to do something useful in space? And part of my research group's interest is in looking at using microorganisms in space exploration. And there's a growing interest in establishing a permanent human presence on the Moon and Mars. And during the last 4 years, my lab has flown experiments to the International Space Station to look and see how microorganisms can be used to extract ions again from rocks, things like rare earth elements. And we do this using a process called biological mining or biomining. This is not science fiction. On Earth today, about 25% of the world's copper is extracted from rocks using biomining. What you do is you dig up a bunch of rocks, put them in a giant pile, you pass water through, the microbes grow, and the microbes eat the rocks, and they release the elements which you can then purify and extract. So this is a process called biomining. Can we do biomining on the Moon and Mars? Well, the first thing you might ask is, why would the effects of space or gravity influence microbial behavior in space? So this is what I'm talking about, gravity. Microorganisms are too small to detect gravity directly, but fluid dynamics is changed in gravity. So in microgravity rather. In microgravity, there is no sedimentation and there is no convection because there's no density differences to drive convection. This is some work that was done by another group, just to show you that on the left-hand side, on Earth, this microorganism grew these mushroom-shaped structures, and on the right-hand side, in that red ellipse, in microgravity in space, it formed these sheets of microbes. So this is just simply to show you that although microbes cannot detect gravity directly, fluid dynamics is altered, and this subsequently affects the growth of microorganisms. So it's an indirect effect. So what we wanted to test was, would these changes, similar perhaps to this in space, alter the ability of microbes to do something useful? So we looked at the extraction of rare earth elements on the right-hand side from basalt. Basalt is a common volcanic rock. It's found on Mars, it's found on the Moon. In those two images, you're just looking at plains of volcanic basalt rock. So this is the material that we chose. We chose to look at rare earth elements because these are elements used in high technology. There's a great deal of interest in purifying them and recycling them. They are of use because they have magnetic and fluorescent properties. They're in your phones. Everyone sitting in this lecture theatre has rare earth elements in your phones or your computers. They're used in defense applications. So they find applications everywhere, and they will be useful in space. So we spent 10 years building a miniature biomining reactor with the European Space Agency. This is the reactor. It contains basalt rock, and on that rock are our microorganisms. We had 3 species. And what happens is these things get flown into space. They get put in a centrifuge to simulate Martian gravity. This is Luca Parmitano, who is the astronaut on the space station assigned to look after our experiment. Once they are in the apparatus, they are triggered from Earth. They are given their food, which injects into the container. And then 2 weeks later, we inject a fixative that kills the microbes and stops the experiment. And then we fly them back to Earth and we see what's going on. These are holiday photographs, not science, but after you've spent 10 years planning an experiment, it's very exciting to get to the final point of launching it. So this is us in Florida at Cape Kennedy, preparing our experiment at the International Space Station processing facility, which is next to the launch pad. And then we launched it in commercial resupply mission 18 with SpaceX. And this is our capsule with our microbe approaching the space station. There was a lovely day. One day after our experiment went to space station, we were sitting in a garden hosting a barbecue for the people who had helped us at the space station assembly facility. And the International Space Station went overhead in the night sky as a little bright dot. So we were able to sit in a garden and watch our experiment going over in the sky. And that was quite exciting. So what did we see? Let me just get to the point here. On the right-hand side is a graph. On the x-axis is the rare earth elements, and on the y-axis is the increase in the release of elements in the biological experiment compared to the non-biological controls, the percentage increase caused by the microorganisms. And what you can see very simply is in that red box, microorganisms have been able to increase the leaching of the elements from the rock by several 100%, between about 100, 400%. It varies according to the rare earth elements. But what's interesting about this experiment is we found no significant difference between the different gravity regimes, the microgravity, the Martian gravity, and the Earth gravity. This was a surprise, given what I said to you earlier about gravity affecting fluid dynamics, which should then affect the microbes. So why did we see no difference between the gravity? Well, on the right-hand side, and it's rather a lot of data, but all it shows you is the different gravities: microgravity, Mars gravity, flight 1G, simulated 1 gravity in space, and real gravity on Earth. And this is the number of cells, the number of bacteria in the containers. And very simply, what happens after 2 weeks of experiment, the number of cells in the container had become the same regardless of the gravity conditions. So although gravity may well have affected the growth of the microbes, and we would predict that's the case, if you leave it for long enough, you get enough microbes, you reach a carrying capacity, stationary phase, where there are enough microbes to do leaching. So we think that what happened is that regardless of the gravity, independent of the gravity, we ended up with the same number of microbes that then did biomining and leached the elements. And this is why we saw no statistical significant difference between the different gravity regimes. So you could argue that that's good news. That means if you want to do biomining in space, gravity doesn't matter. But of course, you should be careful because our little experiment is 5 milliliters of fluid. We're not talking about an industrial scale reactor where gravity might have a bigger effect. So I would personally be quite careful in using these data to make real predictions on industrial processes. But nevertheless, it was the first biomining experiment in space, and it demonstrated the possibility of using microorganisms to do biological processes in space. We also repeated this experiment using asteroid material. 2 years ago, we did an experiment on space station where we demonstrated the use of microorganisms to extract platinum group elements from asteroidal material. And these are also valuable elements of use in industry. And I won't talk about the data, but this is leaching data and various geological data on how these microbes interacted with asteroids. So we used meteorite, L-chondrite, which is a common type of meteorite in the solar system. We took some of that material and we sent it back up to the space station. Actually, probably one of the rare instances in which a piece of asteroid has gone back up into space, having landed on Earth. So it went back up into this experiment. Finally, I just wanted to present a thought. A few years ago, I was asked to, in 2022, I was asked to write a paper on the future of microbiology in space and what I imagined might happen. And this is what I call a muncher. It's covered up by the thing at the top, but it's a machine about the size of a dog, and it walks around on an asteroid. On the right-hand side are 2 images. I should declare that I used artificial intelligence to make these 2 images. So the students among you should, I mean, artificial intelligence is terrible for learning, but the one good thing it does is make images of concepts and ideas in your head. I actually find it quite useful for this. So here's a muncher. It's walking around on an asteroid. It crunches up the rocks. It feeds them this material into bioreactors. And in those bioreactors, microorganisms are turning the material into metals or plastics or pharmaceuticals. And then the material gets deposited and a robot or a human can come along and collect this material. And the idea is you could make thousands of these machines and send them to the solar system, and they would go to the Kuiper belt or the Oort cloud and simply start munching through all those rocks and turning them into useful products. And maybe you could even make a muncher that would extract elements and produce a copy of itself. So these things would self-replicate and essentially eat the solar system using microbes and turn it into useful products. So while we sit here thinking about astrobiology and going about our lives, the munchers would be producing useful materials for civilization, either here on Earth or out in space. You will appreciate that there are interesting philosophical and social questions, which I'm not going to address here. Is it ethically acceptable to send 1 million munchers into the solar system to start eating asteroids and planets and turning it into useful products? I think the ethics people have something to say about this. And whenever I present this idea, I get these horrific looks from, you know, social scientists and philosophers. But anyway, this is technologically where we are. This is not, I mean, this is science fiction, but there's no reason why you couldn't do this. And I've had conversations with engineers in Edinburgh about building a prototype muncher. And in the next 100 years, 200 years, there's no reason why microbial biotechnology and space technology will not be in a position where we could do things like this. So the social scientists will have some work to do to decide whether they will let us do this. But anyway, that's just to give you an idea of how a physical understanding of the way in which microbes operate in extreme environments can be useful for space exploration and maybe ultimately space settlement. So I hope that's given you some idea of some of my areas of interest, some of the things I do, the connections between microbiology, astronomy, and physics. I want to thank these people who supported my work in the UK, but of course I especially want to thank the Fund of the President of Ukraine for supporting my visit here this month. And we've had all sorts of fantastic discussions, already set up new PhD collaborations. It looks like I'm going to be doing, the Vice Rector has asked me to do an astrobiology course starting in September for students at KNU, and there's all sorts of other things we're talking about. So this has already been a very productive visit here. And as I say, I'm around still for another 2.5 weeks. So if anyone wants to talk about you know, eating the solar system with microbes or anything else, just let Ludmila know, and we can have a discussion. So thank you very much.
Шановний Чарльзе, ми щиро вдячні вам за вашу прекрасну доповідь. А якщо ви не проти, то ми зараз візьмемо такий режим. Катя, будь ласка, підійди сюди. Для того, щоб простіше спілкуватись, ми можемо задавати вам питання українською. Катя буде то зали будуть питання українською. Катя буде вам перекладати на англійську, і ви відповідаєте, Катя перекладає. Прошу, які питання є? У кого які є питання? Так, прошу вас. Ти чуєш? Чекайте, погано чутно.
Голосніше, будь ласка. In your presentation, you have mentioned that in your study you focused on the influence of microgravity on the microbes.
Yes.
Which are the influences who have isolated the study, which can become subjects of the following studies for these biological minute microbes in space?
Yeah, this is an excellent question. Thank you. I think there are two things that would be useful to do. One of the problems with our experiments is it was an endpoint. So we did the experiment and we looked right at the end after 2 weeks. It would be really useful to know what had happened in between. Because as I mentioned, we think that growth was changed by gravity, and then eventually we had enough microbes to do the biomining. So the hypothesis would be there was an effect of gravity earlier, but not later. And the only way to answer this is to look at the experiment right the way through the time course. This was an experiment that was sent to space station, it worked, and then it was sent back to Earth. So we were not able to take time points because we didn't have, you know, it's not possible to do that. So this would be one thing. In terms of the scientific expansion of the experiment, I think it would be interesting to look at radiation effects. the International Space Station is below the Van Allen belts. So the radiation levels are actually, I mean, they're higher than the surface of the Earth. They're not the same as interplanetary space. They're quite low. And of course, if you were doing biomining on the moon or on an asteroid, you would have much higher levels of radiation. In some sense, that's a good thing because it was one variable removed from our experiment and we were essentially just looking at gravity. So the experiment is simplified and that makes it scientifically an easier and probably better experiment to do. But ultimately, if you were to try and do this in a real environment in space, anywhere beyond Earth orbit, you would need to know what the effects of radiation were on microbial growth and possible interactions with rocks. So I would do this experiment on the moon. And on that score, we did actually have a conversation with a company 3 months ago, a commercial company that's planning to send robots to the moon, about whether we could send our experiment there. The problem is you can't get it back again. So you have to measure everything in place and you can't get the rocks back, which for us is a problem because we need to look at what the microbes are doing. So really what we need is a laboratory on the moon, and then we can go and redo our experiment in a more realistic environment. So that's what we need. And if anyone's got any good ideas for how to put a laboratory on the moon, this would be good.
Thank you. Будь ласка, ще питання?
Yeah.
You told that the soil on Mars is unfavorable for life because of high ionic strength, and high ionic strength because of multivalent ions. But ionic strength depends also on concentration of these ions.
Yeah.
And we don't know what concentration it was because we don't know how much water it
Yes.
So
yeah, this is an excellent point. So maybe I was not clear and I should have been clearer. I certainly wasn't saying that all of Mars is uninhabitable because of these salts. What I was saying is that there are many of these salts on Mars in different locations and in the subsurface, and where they are concentrated by evaporation and they get to a concentration where they would limit life, that limit would be caused by ionic strength as well as water activity. But it's certainly the case if it's dilute enough, it will not limit life. You know, a dilute solution of magnesium sulfate is perfectly okay. You can grow microorganisms. It's just simply that where you come up to the limits, those limits will be defined by ionic strength as well as water activity. So it's more of a comment on what actually sets the ultimate limits for life, not that all life on Mars would be limited by ionic strength. Clearly, it would depend on where those salts are and their concentrations. But this is a good point, and maybe I should have been clearer. Does that clarify your... Yeah. Thank you.
Ви можете задавати питання українською мовою, хто бажає. Є перекладач, будь ласка.
I actually have a question.
Sure.
So you said that the number of life-limiting factors is large, right?
Yeah.
What's the most exotic one that you are aware of?
Most exotic one.
Yeah.
That's interesting. I think one of the most exotic and interesting, I mean, it's not too exotic, but I think it's interesting, is liquid water under extremely high pressure. So in the subsurface of the Earth, there's a lot of liquid water, but at high pressure. And because it's at high pressure, it's also sometimes at high temperature. So the idea that you can have, so water under different pressure and temperature conditions is interesting. And in my lab, for example, I mean, just go to the other extreme. We've had insects swimming around, fish swimming around in boiling water. So if you take water and you put it in a Mars chamber and you reduce the pressure, of course, it will start to boil. And it's just amazing to look in a Mars chamber at creatures swimming around in water that's boiling. And it's sort of a crazy thing. I mean, physically, there's nothing particularly astonishing about it. It's just boiling because it's hit the triple point, but the water's there. But it's exotic in the sense that the human brain, you're not used to seeing life swimming around in boiling water. So there are these strange things that you can observe in the lab that are very interesting when you start to change pressure and temperature. And I always think that's interesting. In terms of high pressure, the fact that you can have vast quantities of liquid water that is highly pressurized at high temperatures, which are well above the upper temperature limit for life. And yet this liquid water cannot support life. And often we're used to this idea that where there is water, there is life. And that's because, of course, we live on the surface of the Earth. And wherever we see liquid water in a lake or a river, we generally see life. So we're not used to this idea that actually there are large volumes of water, particularly beneath the surface of the earth, that are unsuitable for life. So where there is water, there is not liquid water, there is not necessarily life. And that, again, not necessarily exotic, but it's an interesting combination of pressure and temperature that creates counterintuitive views about where life is and isn't. That's a great question. Yeah.
Прошу, ще які питання? Будь ласка, пані Анна.
Charles, thank you very much. Probably I'm only biologist in this room. My question will be related to your biological experiment. You showed on the screen there you compare the number of bacteria which grow in the solution with the sodium chloride in the multi-valent solution. For me as a biologist, interested how did you pick up the list of bacteria which you grow in that solution? So did you search for possible bacteria which possibly can grow in this?
Yeah, this is a great point. We used salts from the edges of a variety of different salt pans and mixed them together to create an inoculant that we then introduced into the flask. So this is where you see no growth of microorganisms, you have to be quite careful, because you could argue, well, just maybe you haven't introduced the right organism and nothing has grown. But it's really difficult to get beyond this problem. You have to add something. And if you're trying a brine that is a very sort of alien chemistry, well, maybe nothing has evolved to live in there. So why would you be surprised? So this is a good question. But having said that, what's interesting is if you do add microorganisms into these brines, you definitely see clear physical limits, like I've described, of water activity, which is very consistent across a variety of environments on Earth. And in the case of those Martian brines, never once could we get anything to grow in a solution that had an ionic strength of more than about 10 molar. And that suggests, there may be a microbe out there that will grow at 15 molar. I wouldn't rule this out. But it was interesting that this always seemed to be a limit. And it suggests to me that generally, once you get about 10 molar ionic strength in a solution, the charge density, at least with life that we know it, with the biochemistry of life that we have on Earth, this becomes a challenge to the folding of molecules and the operation of enzymes inside the cell. But you should always keep an open mind, because maybe there's other limits.
Okay. Thank you very much. And maybe it's like more or less philosophical question.
Yeah.
From your presentation, my impression that using your experiment model system, it's more about to show that in current situation, it's no possible life outside of the Earth. Like you show the experiment, like model experiment on Mars and so on. So like in your future experiment, what is your main idea? Would you like to find the possible bacteria which can grow and it will be like experiment can be the life outside of the Earth? Can you explain a little bit?
Yeah, thank you. This is a good question. I tend as a scientist to try and avoid too much speculation about whether there's life on Mars or not. I try and stick to, if there was life, what physics would limit it? Because for me, that's a fundamental question. It's interesting, and it's independent of whether there happens to be life on Mars. Has the geological history of Mars and the types of chemistry you find there, how would that limit life if life was to exist on this planet? That's a fundamental question about the limits of biology, and it bears no relation to whether there happens to be life on Mars. It goes back to this question earlier that, I'm not saying there isn't life on Mars, because you can just have a dilute salt solution and something will grow in it. The question I'm asking is, when you go to the very limits, what are the physical principles that limit life? And in the case of Mars, these multivalent ions create this limit, this ionic strength limit that I find interesting because it's very rare on Earth. So that's the interest. Of course, as a biologist, I'm interested in looking for life on Mars. I'd be very excited if someone could bring me a sample of Martian life, because then I could do lots of interesting experiments in the lab. But you know, this is for the future, and it's for other people building rovers and planning human missions to answer this question. But having said that, the work that we do can help this. Like I showed with the craters, you know, the work on ionic strength would suggest that going and digging in some iron sulfate on Mars would not be a good place to go and look for life, because it's probably limiting. You probably don't want to go and dig into some, you know, magnesium sulfate and iron sulfate on Mars. You should go somewhere where there were not so many salts. So there are implications of what I've discussed for how you might look for life on Mars. In terms of where I go in the future, I continue to be interested in any sort of physical and chemical experiments that can yield insights into the limits of this replicating, evolving matter we call life. I'm interested in the practical applications of this for space exploration and settlement. It just happens to be something I'm very enthusiastic about. Humans moving beyond Earth, it's personal interest, and I usually link that to biology. And you know, and as a side interest, I'm always interested in trying to apply what I've learned to thinking about how you would look for life elsewhere. So I don't know whether that clarifies what your question. Thank you.
Прошу, ваше питання.
Pressure types and
Не чутно, голосніше.
the ammonia. Maybe could you please briefly elaborate on the effects of ammonia and pressure?
Oh yeah, so there we were looking at, so when you add ammonia to water, you get very high pH solution. And often in the literature, you'll see that life cannot grow in high ammonia because the pH is 12 or 13, and this is no good for life. And we were interested in whether ammonia exerts a limit to life based on pH, or whether there's an independent effect caused by the ammonia molecule. And you can do some nice controlled experiments, which is what one of my PhD students did, where you can look at the cell response to high pH by using non-ammonia solutions at high pH, looking at the biochemical and metabolic effects of those high pH solutions, and then comparing them to solutions of ammonia of exactly the same pH. And what you see is that the effects are not the same. The ammonia molecule is disrupting biochemistry independent of the pH. And we think it's probably something to do with the hydrogen bonding of the ammonia that's disrupting hydrogen bonding around proteins of water, the water hydrogen bonding. So the ammonia is affecting maybe the structure of water, or it's getting into the active site of enzymes and causing bonding in those active sites that's disrupting the normal function. And this is not an effect of pH. So this was just one experiment we did. In terms of pressure, our work was really looking at whether you could reverse the effects of pressure by using salts that cause disordering of molecules. And Stuart Galt, who's a PhD student in my lab, is now finished. What he managed to show is that if you pressurize cells up to about 5 kilobar, so, you know, 3 or 4 kilometers deep in the crust, the pressure will cause molecules to reduce in volume, and it will reduce enzyme activity. If you then add in perchlorate salts, which are disordering, if you add them in at just the right concentration, you can reverse that pressure effect. So the pressure causes the molecule to collapse. And if you add a small amount of perchlorate, it will disorder, but disorder just enough to provide activity in the enzyme again. In other words, these disordering salts that look very damaging to life can reverse the damaging effects of pressure and become good for life. So that was the work on pressure.
Дякую. Прошу ваше питання.
Your lecture focuses on the outer limits of life.
Yes.
What about looking at those limits from within? What about looking and understanding what exactly breaks at those limits?
What exactly?
What exactly breaks in the organism itself at those limits?
Oh yeah. So that's very much part of the the work that we try and do. So when I say the outer limits to life, someone also raised this yesterday. It's a really good question to say, why don't you look at the inner limits to life, like, low radiation? I should say the outer, this is just what. So when I say outer limit, that could be low radiation or high radiation or low temperature or high pressure. So when I say the outer limits, it could be low concentrations of something or high I'm simply talking about limits. But in terms of what's going on inside the cell, absolutely. So we study the proteins, we study the metabolites, metabolomics, proteomics, we study the genetic responses that are going on in the cells. We're very much interested in understanding at the molecular level what is happening inside the cell at these limits. So looking at survival is just the initial step. You then want to know what is it that's actually not functioning inside the cell, as you put it, what's broken. So we do that as well. Today, I sort of focused more on the physical side, just because I thought this would be more interesting for this audience. But there's also a lot of sort of very detailed molecular data. A lot of it is not necessarily that interesting, but there's a lot of molecular data on which proteins are being changed in these different extremes. And that can provide some explanation of exactly what the target of dysfunction is inside the cell. That's a good point.
Ще є питання, прошу ваше питання.
About searching for life active on exoplanets. So, what is a limited spectra, or the maximum parameters of you can measure on the exoplanets to be determined to say something about life forms?
Yeah, that's a great question. I think the first step, I think the first thing you should look for is liquid water, which is what astronomers do, right, when they look for the habitable zone around a star. Because although I said you can have liquid water that is unsuitable for life if it's at too high temperature, too high pressure, without water, as far as we know, life is not possible. So you can have a planet with low concentrations of ions and energy source, carbon and nitrogen and phosphorus and all these things you build a cell from. But if there's no liquid water, there's no possibility of doing anything. So I think it's a reasonable first step to look for a planet that looks like it has liquid water. Probably if I was to take the next step, the next obvious thing to look at is temperature. So if the temperature is very high, 200, 300 degrees, maybe it's high pressure on the surface, so there's liquid water, but the temperature is very high, I think that would not be suitable for life. So you want liquid water and a suitable temperature range. Once you have that, then I would worry about ions and other aspects of radiation and these other extremes that define the limits to life. But water and temperature, if there's no water and the temperatures are too extreme, then I think everything else is in some sense a little bit irrelevant. So I would start there.
We can measure lots of parameters about exoplanets. We can measure the luminosity of the host star, and we can measure the distance between planet and star. So we can assume we can determine the temperature on the planet as well as another parameters like from a spectral characteristic, effective acceleration of gravity and so on. And we have like data from that. But what's next? What we are looking for? So with spectrum, maybe we can maybe we can see organics in spectrum. However, on comets we can also find out the organics, but it's not the factors that can influence their silicon forms. So what is the next thing?
Yeah, this is a great question. I mean, from a biologist's point of view, I would really like to know really a lot more detail about what's going on the surface. In other words, the composition of the planet. So you as an astronomer, I guess what would be really useful is the planets where you've got the transit method, so you've got the diameter, and maybe the Doppler shift method, so you've got the mass, and then you have a density. And once you have a density, then the geologist can tell you something about what it might be made from. And then the biologist will find this useful, because once you have some idea of what it's made from, we can think about what ions might be there, what the liquid water might be like. Is it going to be like ocean water, and what the composition might be like. I think what a biologist just wants to know as much as possible, but a biologist wants to know, is there liquid water? Is there an energy source as well? Is there a disequilibrium that will provide energy? And that itself is dependent on knowing something about different chemical compounds. So, I mean, just for example, you and I, we use organic material from our pizza at lunch and we burn it in oxygen, and that's a chemical disequilibrium. So what the astronomers need to do with the chemists is provide a list of these different chemical compounds and possible energy disequilibria. And then as a biologist, I want to know about some of the things I've discussed today. What sort of ions might there be? What might be in the liquid water? Would it limit life? Is it a suitable environment for life? So I would say, maybe this isn't a very helpful thing to say, but because it's sort of obvious, but I suppose for a biologist, we just want to know a lot more about the chemistry. We want to know what that surface is actually like, what energy sources are available, and what the chemistry of fluids would be like. So the more information we can get about that, the better. And probably some of that information can come from density calculations, where you can start to predict the geology. If you have a density of an exoplanet, and something about, where it is, how distant it is from a star, maybe you can say something about whether it's a terrestrial rocky planet. And then you can start to make some assumptions about what the surface of that planet might be made from. And then it's quite an easy matter for a geochemist to take that composition and model interactions with water and work out possible geochemistry. And then they can say to the biologist, well, if this planet is made from this or this, in these cases, if you mix this with water, these are the fluids you'll have. And then a biologist can start to think about, is this a good environment for known life?
Ще питання є? Прошу ваше питання.
Some, but as far as I understand, what we're trying to see is regarding research and Mars. What we try to see if microorganism from Earth that fit Earth's conditions and create the conditions from the Mars. But if we try to artificially install microorganisms on Mars that fit specifically for the Mars, we try to create the organisms that specifically fit the conditions on the Mars. Why?
We can do this. I have a colleague, Erica de Benedictus, who I've published some work with. She's now in the US, and she's just set up a company called Pioneer Laboratories to try and genetically engineer microbes that will survive in Martian conditions, specifically for the purpose of trying to introduce them into the Martian environment. This, again, has interesting ethical implications. So right now, there are rules called planetary protection rules. They're not international law. They're regulations agreed by the Committee on Space Research. But most space agencies adhere to these rules. And Mars is a place that has been determined to be a place where you cannot artificially introduce life, okay? You can't introduce microorganisms. Well, as I say, it's not international law, but all countries that have a space program agree to abide by these recommendations from the Committee on Space Research. So no one has yet proposed a serious experiment to deliberately introduce microorganisms onto Mars. And I should also say, it's a place where it would be very difficult to do biomining. So the things I was talking about today, if you went to a space agency now and said, I want to take a 1,000-liter fermentation reactor to Mars with microbes in it and do biomining, there would be an international outcry. So this is not happening anytime soon. The interesting, I guess, it's not a scientific question, it's a policy question. The interesting question is, how far would you have to demonstrate a lack of life on Mars before you are ready to say, okay, people can take microbes to Mars? Because you could always argue, maybe there's microbes 2 kilometers underground in some little oasis. How can we ever prove there's no life on Mars? This is a very difficult thing to do. And so maybe you can never reach a point where you're 100% certain about a lack of life before you allow people to do these sorts of experiments in introducing microbes. That is not a science question. It's a political and policy decision at some level of confidence that other people spend their time thinking about. But it's a really interesting question, because I suspect at some point we have to decide about this. One final point about this, it's impossible to send humans to Mars without contaminating the surface of Mars. Human beings shed microbes all the time. You cannot build a spacesuit that will not shed microorganisms. This is just an engineering fact of life, that if you put humans on Mars, there will be microbes on the Martian surface. Some people say human explorers should not go to Mars until we sent more robots and demonstrated a lack of life. Some people say it's okay to send humans because even if they shed microbes, they're probably not going to survive and they're not going to affect any indigenous life. And that is more of a scientific question about the survival of microbes on Mars.
Makes it so, I guess, bad for ecology is that people do this. So,
Yeah.
so why is it so bad for ecology?
Because there's still a lot we don't know about what happens if you introduce microbes into an ecosystem. There's a great deal of ecological data at larger scale of introducing rats and other organisms onto islands that cause a lot of damage to local ecosystems. These effects are less understood with microorganisms, but just like with large animals, it's quite possible that if you went to Mars and introduced lots of organisms and there was life on Mars, it would disrupt those ecosystems. And the reality is we actually don't know a lot about this with microorganisms. We know that you can introduce rats and things into islands and they cause disruption. Plants that grow very rapidly. Microbes, this is not so well known. So I think we do need to be careful. We do need to think about this.
Ваше останнє питання.
Так.
I would like to ask you, are you involved in
Можна голосніше сюди, пане?
are you involved in any projects of studying water on Jupiter's satellite Europe? And is there any projects like that?
Yeah, there are. I'm not directly involved. I had a student who did some work on looking at we tried to simulate the plumes of Enceladus, and we put microbes in a machine that made plumes, and we were trying to see whether the microbes would survive coming out of Enceladus plume. And this was like a 6-month experiment. This was quite fun to do. I don't work so much on the outer moons of the solar system. There's only so many planets you can work on, and it would be nice to do more research on some of these environments. But the icy moons are very interesting. Low temperatures, high pressures, possibly high salts as well. So some of the things I've talked about today have applications to the oceans inside icy moons as well as surface of Mars. So certainly you can transfer this knowledge. I just haven't done so much on icy moons, but there are research groups trying to simulate and trying to model the composition of the oceans in Europa and Enceladus and other icy worlds. Yeah.
Шановний Чарльзе, ще є питання в чаті до вас. Катя, почитай, будь ласка.
The question in the chat, but it's in English. There's an opinion that in the observing area there are only 2 to 3 star systems suitable for life existing, and this system are anomalously far from us. Is that true? Yeah.
Well, this comes back to the question earlier about what you look for on a planet to determine whether it's habitable. So there are lots of planets within tens to hundreds of light years that people believe might be habitable. There's catalogues of habitable planets. But when we say catalogues of habitable planets, habitable in that they might support liquid water, might have the right temperatures. But could they really support life? We don't have enough data yet. But they are, tens to hundreds of light years away. So, there's no prospect in the near term of visiting these planets. And that in itself is an interesting discovery, that habitable planets, if they exist, are going to be a long way away. We will probably not sample them directly. But what is exciting is there are lots of candidate planets that might be habitable. What we do know now is that there are many Earth mass or roughly Earth mass planets orbiting other stars. And that, of course, was not known 10, 20 years ago. And that's extraordinarily exciting because it shows that if we build the right telescopes, there is an enormous amount of work to be done in detecting the spectra of biosignature gases and habitability. And maybe, in 100 years' time, on the 200th anniversary of Vernadsky's book, The Biosphere, people will come into a lecture theatre like this, and either we will know that there is no evidence for life, no obvious evidence for life in our neighborhood, which will not tell us there's no life in the universe, but will tell us, at least locally, there is apparently no life, which could suggest that life is very rare. Or people will be sitting in this lecture theatre looking at slides of known planets where there's high concentrations of oxygen and biosignatures. And the question, is there life elsewhere in the universe, will be answered. And it will be now more of a question about establishing the numbers of these planets and statistical information about their abundance rather than whether life exists. And I think that is a really exciting, thrilling possibility for the next 100 years.
Шановні колеги, ми щиро вдячні професору Чарльзу Кокеллу за дуже приємну змістовну доповідь. І крім того, я думаю, що ми всі подякуємо ще усьому британському народу за ту підтримку, яку він кожен день дає нам в боротьбі за нашу незалежність і за нашу свободу. Дякуємо тобі, Чарльзе. Дякую всім. Дякую. Наступний наш семінар буде присвячений теж астрономії і буде доповідати член-кореспондент нашої Академії наук, яка стане скоро директором Астрофізичної обсерваторії України. Дякую.